And our first talk is from Yuki Yamagata. And she's from Japan. And oh, I forgot to say that, you know, all the three talks before the keynote speech is about the emerging topic on COVID-19. And Yuki's talk is about a new ontology called Homeostasis Imbalance process ontology. And she's going to focus on a study on COVID-19 efficiency process. Jago, could you play the video for her?
Sure.
Hello everyone. Today I'd like to talk about knowledge systematization of COVID-19 infectious processes and developing homeostatic imbalance process ontology HoIP. Background. One of the major issues of COVID-19 is to decrease the severity of disease. For example, ARDS. We need a better understanding of how biological system functions against SARS-CoV-2. So our objective is knowledge systematization of COVID-19 infectious process based on ontology. COVID-19 infectious mechanisms are complicated, so we require various expert knowledge. But experts haven't shared knowledge, so we need to picture the whole knowledge interdisciplinarily. We focus on homeostasis imbalance process, and by developing HoIP ontology, we tackle these issues. Issue 1. Concerning COVID-19, there are a wide variety of pathological processes and symptoms. No symptoms, fever, dyspnea, and so on. So we need a unified representation framework to represent processes from no-symptom to severe clinical manifestation. Several pathway DBs provide COVID-19 pathways such as disease map, CAC, and so on. However, they mainly focus on molecule-molecule interactions. To understand mechanisms, we need to capture process-process interactions. So this study defines a COVID-19 cause as a series of processes in an organism, from viral attachment to clinical manifestation. And in our representation framework, we represent infectious cause as causal relationships of processes. And more, we introduce imbalance theory. In the imbalance model, basic units are functional supply as biological defense, functional demand as viral action, viral balance, imbalance, and outcome. And we also focus on inflammatory response. And we describe inflammatory response as the result of the immune response against viral action. This slide shows imbalance model. In mild cause, immune defense and viral actions are balanced. And the upper right shows inner right. When immune defense becomes very low by viruses, such as type 1 interferon inhibition, an imbalance occurs, which brings about establishment of viral infection. And since innate immunity response becomes low, so no inflammation occurs. Please look at the lower figure. We can distinguish two severe patterns. One is excessive attack by virus, and the other is excessive defense. When innate immune defense very high, which leads to high inflammation and causes cytokine storm and so on. So our imbalance model can distinguish viral action and immune response, and support to capture various inflammatory responses. So our approach will contribute to new drug development. Issue 2. Various granularities in the body. In the host body, processes are highly diverse and various granularities from molecule to organ, so we need to represent them uniformly. So we focus on functioning process and introduce a functioning decomposition approach. We decompose functioning of one system into subfunctioning of its parts. For instance, concerning removing infected cell, to achieve this function, early removing infected cell in innate immunity and removing infected cell in adaptive immunity work as subfunctioning processes. And early removing infected cell in innate immunity is further decomposed into three: NK cell-mediated apoptosis, phagocytosis, cytolysis, and we describe the participants, such as macrophages, neutrophil, dendritic cell, as participant. In this way, this framework can cover across granularities systematically. And we can also decompose the viral functional demand process uniformly. Issue 3. Knowledge systematization. We develop HoIP ontology with three-layer structure in the same manner of the toxic process ontology, TXPO. Top layer, domain, intermediate layer, and lower layer. The top layer domain is domain neutral, which defines very basic and general terms, referring to BFO. We also define functioning terms, for example, transmitting, transporting, which are used not only in biomedicine but also machinery. The intermediate layer is biomedicine dependent, and we reuse ontologies such as GO, PATE, HPO, vaccine ontology, and so on. And in the lower layer is homeostatic imbalance dependent, which includes ER stress, fatigue, refinement from TXPO, and we add COVID-19 cause and its specific processes. And other disease causes such as diabetes are also defined. This slide shows example of specialization of COVID-19 infectious cause. Easy relationship between causes is shown as inclusion relationship. For example, severe COVID-19 cause includes all COVID-19 cause nodes, and which are specialized into very high. And moreover, we can see an added new process in downstream, positive regulation of blood coagulation. And in COVID-19 associated with ARDS, lung-specific processes such as alveolar epithelial cell injury are added downstream. We use protege for ontology development, but it's not easy for understanding for COVID-19 researchers. So we are developing visualization system. LEAVES is an ontology-based biomedical knowledge navigator system, which is extension of ToxPilot. It provides course map, process map, path search, and ontology-based information. This shows the system architecture. details I'd like to skip. Cause map is a visualization map of causal relationships between processes. It shows causal relationships, and also we can see both human and viral molecules. Based on HoIP ontology's information, we can see how each vaccine relates to the specific processes. For example, many vaccines, including messenger RNA vaccine, participate in immunoglobulin-mediated neutralization of the S1 protein receptor binding domain. And by following upstream, we can see why symptoms can occur. And also, we visualize severe symptoms and disease. For example, in the COVID-19 with ARDS, we can see dyspnea as a symptom of respiratory dysfunction. Here is a passage screen. From now on, we show how our model contributes to finding potential risk across multiple disease causes. In the intermediate layer, we can find common processes across causes. For instance, microvascular dysfunction common to COVID-19 and type 2 diabetes. So here is a visualization of the general cause map in leaves. Using common process, microvascular dysfunction, we can combine and produce general causal relationships. So this map can show both specific and common processes across multiple causes. So we can find common factors and new risks beyond causes. Furthermore, by general causal relationships, we can see lopinavir and ritonavir are upstream of hyperglycemia, which might have potential risks because crosstalk among ER stress, COVID-19, and diabetes. So our approach contributes to finding potential risk factors and explaining how factors affect the functioning process downstream. Here beyond domains, by linking isolated data on ontology, we can show whole picture of the knowledge. In diagnosis, from findings, we can trace back to the fundamental cause. Now we have started developing a working group to harmonize 7 ontologies for knowledge interoperability. HoIP is open to the public on BioPortal and GitHub. Please access them. Thank you very much.
Thanks, Yuki, for the next presentation. So just wonder if anybody has any questions for Yuki.
I have one question. This is Anna Maria. And I would like to know how you are going to integrate, or if you're thinking to integrate all this that is really very nice with the approach that has been done to the N3C database.
Yes. Sorry, ask again.
There is this collection of data through, I think is the word, collection of data about COVID on N3C study.
Yes.
And there is they have this kind of approach, ontological approach, I think using more a mondo ontology for the clinical part. And I was thinking if you have think about a way these two efforts could be integrated, since you are more looking at the biological process and they are
Yes.
looking more, yeah, the clinical part. Yeah, that.
In future, I'd like to more integrated like Mondo or the ontologies. So, but now I have just started now. So I collaborate with Sido and comparing the both terms are same or much close. So now I'm analyzing it.
Okay, perfect. But it's nice work.
Thank you.
Yeah, Yuki, it's nice work. I can ask you one question.
Yes.
So, for the your ontology, since have used many processes, right? So the gene ontology has some related, I think,
Yes.
processes. Like, are you kind of aligned with them, or
Yes.
you are developing your new set of terms for
Yes.
your own purposes? Yeah.
We first I search the geoterm as how can this geoterm is fitting to our ontology or not. So then select the geoterm is can be a parent of our terms or not. I checked, and then if they are not but geoterm is only normal biological biological functioning. So if they are not enough, I will add other terms.
Okay. Yeah. Thank you.
Any other questions? Well, I have two questions. So one is, so I think this ontology is very important, but with the emerging, progress on related to COVID-19 studies. So I just want to try. So my assumption is the current ontology is generated manually, right?
Yes.
So do you have any plan, maybe make it like more automatic or semi-automatic, maybe use natural language processing methods?
Yes. So first, I developed. I created manually, but if the term is more making more rich, we change. We should shift to semi-automatic with such as natural language processing. And other technology by using it. So I should shift them.
I see. Thank you. And another question, so relevant is, so how would you evaluate this ontology? So, to make sure the ontology is in good quality or, it is serves the purpose.
Yes, first in logically we checked RDL reasoner such as Helmut or ELK2, and I checked the consistency. And the scientific viewpoint, after 3 months or after, we recheck the PubMed article and whether the terms are correct or not, we checked manually. But actually there are a lot of articles, so we can't check to over all the articles now. So that's a problem now.
I see.
Yes.
All right. Thanks, Yuki.
Thank you.